So I guess I will hope to get out of the way at this point, since I get to have the burden of the last word, and certainly ask my colleagues how they would perhaps, knit together the earlier discussion and this one, but with more of an emphasis on the utility and policy making. So, Kerry?
Thank you, Ellen. First, I just want to mention I'm really pleased that as part of the steering committee that put this meeting together, that the discussions have been really wonderful. I have to really thank everybody for doing that, because this is one of our expectations, to have this great discussion. I think we've seen that come to fruition. So I just want to say thank you all for that. Second thing is, I just give you an idea where I'm coming from to sort of the context for my remarks. I work for the United States Department of Agriculture's Food Safety and Inspection Service, and just for those people out there, we're the people that you kind of know as going into the plants and put the mark of inspection right on the meat. Well, we don't literally put it on the meat anymore. We're in the digital age, I guess, but that's who we are. And what we do at headquarters, we actually support our inspectors about what they should be looking for, what levels they should be looking for, and then we tell them, our laboratories sort of, what are the levels that you should be monitoring for these types of. Essentially, what we're looking at are pathogens and chemicals in our meat, poultry, and egg products that we regulate. And so, to be very honest, the FSIS has been looking mostly at microbial adulteration of our meat and poultry products over the last several years, mainly because of the Jack in the Box E. coli episode in hamburger many years ago. And so we've really looked at pathogens. I have to admit, the microbiome is something right up the pathogen alleyway here. So I'm kind of pleased to see that we're talking a lot about that. But I also have to say is that over the past few years, we're trying to bring rebalance back into how we're looking at more chemicals as residues in our food products. And so I want to kind of talk about that in terms of this microbiome. And I'm going to go right to question 2 first, is how does the microbiome fit into defining risk from environmental exposures? And one of the things that we always are confronted by is that people, when they find out that something, chemical X, is in their food or is in their animals, they come to us and the first thing they ask is, well, can I eat the stuff that I just bought? Or can I eat, the product that comes off of my farm animal? And that's a very, very specific question. You just can't say, well, with uncertainty or, variability, well, maybe or maybe not. You got to just say, well, yes or no. And so we do have to have a little bit of, certainty about how much we can really trust our data and our models. And so what we're trying to do is that over the past few years, I said, since the emphasis has not been on chemicals too much, is we've been doing a lot of, I won't say ad hoc, but more like case-by-case analyses of certain chemicals that come to our attention. And so we're finally figuring out, well, we've got to start putting together a much more concerted generic approach so we can have what we call rapid risk analysis, that we can turn some of these decisions around much more quickly than we're currently doing. So this gets into actually what we're doing is looking at exposure scenarios actually from the farm, from where animals are exposed, how it gets into the animals, how it gets into the products that we get from the animals, how it goes through the processing of those animals into products that we actually eat, and then how humans then finally, how much they eat of this stuff, what are their habits and things like that. And so we're talking about exposure scenarios. So when it kind of gets into the, we had a little discussions yesterday about the exposome coming into play. This is really the exposome really from, from soup to nuts almost. And what we're trying to do is model at very specific points what are the types of inputs that will get into our meat and poultry products. So if you look at the farm animals, we're looking at essentially how much are they drinking that might be contaminated with a particular chemical? How much are they breathing if it's airborne? Or how much is it coming through the soil that they're grazing on? And that might be a first point where the microbiome is important, because one of the things we're trying to figure out is through like PBBK type of modeling is after they've had that ingestion or exposure, how much is actually getting to the meat product that we're going to be taking out of these animals. Does it get excreted totally out so it doesn't really get distributed to the meat or something like that? And that's where the PBBK modeling becomes very important and gives us an idea. And here's where the microbiome, again, comes to play. And I think this is what Tom was talking about yesterday, and I think that is very much what we're very much interested in, the accessibility of the materials, as well as the bioavailability that finally gets into the targets, if you will. And so then once it goes through the process, I'm trying to make a long story short here, once it goes through the processing, you got to talk about dilution factors and concentration factors into the different products we have, such as ground meat or something like that. Eventually we get into the humans and we got to figure out, okay, how much are they eating? Okay, then we look for N. Haynes, for example, talks about, eating habits, how much they eat, in a year, how much is in a serving size. We got to incorporate that into our thinking and our model. And here again, we're talking about now the human microbiome. We're talking, usually what we do is you take a look at the actual ingestion and compare it to an RFD, okay? But it'd be much more enlightening, I guess, if you will, if we can take a look at, how much is actually reaching the target that we're really concerned about that might have some type of sequelae for some type of adverse effect that we might be affected. So that's where the human microbiome would help us, again, in this PBK modeling in the humans, if you will. So you can see at different points there. And I think what Jeanette was talking about earlier is we're looking at the microbiome in cattle. That'll be definitely a data point that we can use to talk about how much is actually going to be in some of the product that we're going to be looking at. So I think that's sort of, the answer to the question too is yes, we think this is going to be very important. It's going to be a very important component of our exposure scenario modeling, if that's what you want to call it. So that was sort of the main point I really wanted to make. Now going back to question 1, I just have a bunch of random points here. I'll just throw them out there for people to discuss. These are in no particular order or anything, but these are things I think some of the researchers might want to just kind of consider when they're doing some of this stuff. But since we look at both chemical and microbial, or essentially pathogen exposures, is there any interplay between the two? Maybe this might be some type of aspect of cumulative risk, for example. For example, can one exposure increase the susceptibility to the other? I haven't really heard much about susceptibility here, but I think that could be a very important component here. Because, say, for example, you get into a chemical, comes in, starts wiping out, some of the microbiome, and all of a sudden a pathogen comes in, which normally would not, be able to catch hold or something like that. All of a sudden, you're now increased susceptibility, even though it's a healthy person, okay? And not even talking about immunocompromised people or something like that. So these are types of different people that we might need to examine, and whether their microbiome is going to be affected by any of these types of interactions. And again, I think Alan's brought this up many times, is about the continuous use of antibiotics in animals, and then they obviously show up in a lot of the products that we look at. And we got to figure out, does that also maybe make shifts a person's susceptibility if they have a continuous low-level exposure to antibiotics, for example, that might be in the product. So essentially, what are the effects of eating food from animals that have been on chronic low-dose antibiotics? That could be something that can be investigated. Now, originally I was interested in this particular topic from sort of a genomics point of view, because originally before this emerging committee under NIH, it was actually an omics committee, if you will. And so I was trying to think about, how we can use some of that stuff. Is obviously the omics stuff is not only useful to sequence the microbiome, but maybe using the omics to examine what is being turned on or off, in a sense. If you're thinking about for chemicals, this is again in terms of talking about the toxicity pathways and targets. This is the Tox 21 approach type of thing. But maybe for an analogous way, thinking for pathogens, can we look at analogous pathways in the microbes, i.e., what genes are present or which genes are turned off and on? For example, are virulence genes present or not? That's very important to us, whether you're going to get infected or not. And also, can you talk about infectivity pathways? I'm just sort of throwing that out there, Can you detect these in the pathogens that are coming in? Or are there certain responses in the microbiome that's already there from the commensals that might be useful to look at from the omics point of view? For example, can the microbiome or an altered microbiome impact the infectivity or the response to a particular pathogen? And the last thing I just want to mention, it's not only worthwhile, I think, looking at the effects on the microbiome itself, but what about the responses in the exposed humans, actually the hosts here? Can we identify, say, early signs such as molecular signs, in humans, such as the toxicity pathways that may be turned on, that might be a sequelae to some adverse effect? But this would apply to, say, a pathogen being infecting that. So maybe we can find some early signs for diagnostic purposes, for example. It says, okay, things are being turned on before you actually, while the person is maybe still asymptomatic or something like that. And you maybe have made some early diagnosis and have some interventions or something like that. So those are just some thoughts on those. I'm not going to answer number 3 because we don't really do clinical and non-clinical types of stuff with at USDA. So thank you.
There are 3 people right in a row who are going to, so why don't you go for it?
I'm Rita Shoni, and I'm with the U.S. EPA's Office of Water, and we'll deal mostly with question 2, albeit in a rather roundabout fashion, as is my wont. A couple of introductory comments. I really found this session to be a complete revelation. This is not a way I have been thinking about humans or the environment, and I've found this to be spectacularly interesting and enlightening. I would love to know the composition of my microbiome. Early in my scientific career, I used to grow up massive carboys of Salmonella typhimurium, so I would get a goo that I could then extract enzymes from, so I'm sure we would find some interesting things still inhabiting me.
Not here, though.
Not here, thank you. My office, the Office of Water, is one of those regulatory agencies, if you will, that has the hubris to regulate microbes. And part of the limitations around any kind of policy making in the federal government, at least in the regulatory agencies, has to do with what's written down in the law, telling you what you must do and what you are permitted to do and what you're not permitted to do. So our philosophy in terms of microorganisms in the Office of Water has generally been that they're the enemy. For example, we are very invested in trying to ensure that people who are using water bodies, the United States, do not pick up diseases in the course of swimming, sailing, sitting on the beach, and so forth. We're very invested in ensuring that, for example, fisheries and shellfish beds are not impacted by pathogens to either those particular organisms or to the humans. For example, we wrote one of our criteria for ambient water to deal with zinc in the water. Zinc at sufficiently high levels will impair the immune systems of mollusks, and the next thing you know, you have a shellfish bed that is infected with Vibrio parahemolyticus, and that is no good to anyone. In terms of drinking water, we go beyond the only good bug is a dead bug. The only good bug is a non-existent bug. Because we're also aware of the fact that dead algae can release extremely potent toxins, and that in the course of treating, for example, a source water for algal infestation, if you will, you can release these things into the drinking water, and we don't even know a great deal about some of these toxins. So that has been a large consideration for us. One of the limitations on EPA is that when we're protecting, if you will, drinking water and ensuring that it is safe for public consumption, we are not allowed to ensure that water is nutritious. For example, we have nothing to say about whether fluoride should be in water or not. We set the limits for how much is bad for you. It's up to other agencies to ensure how much is good for you. So if we're, for example, affecting herd immunity by providing sterile water, that's not part of our problem. We're not allowed to deal with that in any way, shape, or form. We are charged with dealing with things like pharmaceuticals in the water, and this is a huge problem that we're trying to discuss right now. But again, we have limitations. We would not, for example, be able to regulate if we even had data indicating that antibiotics from either farm animals or humans were in fact increasing the number of resistant organisms that humans encounter. We could not regulate on that basis. Instead, we would have to regulate on what are the effects of the antibiotics in the water on humans, period, without considering the microbiome. That's likewise, we could we have not regulated the effluent from combined or concentrated animal feeding operations based on their likelihood of providing microbial contamination. We regulated based on how much nitrogen and phosphorus they put out. You know, in our perfect world, we would not have bugs coming out from CAFOs, nor would we would have totally sterile biosolids. Biosolids are sewage, the solid parts of sewage that are treated so that they can be applied to farmland as fertilizer. In our perfect world, it would be absolutely sterile. We're not interested in the composition of the soil as being amended by these particular nutrient-containing biosolids. Little different perspective now in terms of going back to human health. Question number 2, is the microbiome simply a potential modulator of risk? I take issue with the term simply. Modulators of risk are huge. And this is a lot of the way that I've been listening to the presentations today. I really feel, I take a lot of comfort in considering the microbiome to be another organ. As one who deals with exposure assessment, effects assessment, I can see writing that into a pharmacokinetic model as another compartment, and then I'm happy. And I also think that, to bring out an example, as Ellen was pointing out, for metals, perhaps the microbiome is the major modulator of exposure by comparison to genetic differences and variations. It's not always going to be the case. Disinfectant byproducts, there is an overwhelming effect of different genotypes in terms of the ability to metabolize DBPs, trihalomethane in particular. Something that struck me rather tremendously was the consideration of the changes in the microbiome over life stage. Life stage susceptibility is a major function of the way that we do risk assessment in EPA. And I was particularly struck by how rapidly the neonate becomes like an adult in terms of the microbiome. And then everything seems to go completely to hell in a handbasket after you pass 60. And I find that personally very interesting. But also 65, thank you. Right, we're not elderly. It's a good thing. But I think there is little consideration that has been given to life stages other than neonatal or childhood. And this was a huge revelation to me. My last consideration is looking out over the assembled group in the room is, where is everybody? And I understand that particularly in my agency, which this is not lobbying Congress, just took a 15% hit to the budget, microbiome is not on the radar in terms of policy making. And truth to tell, anything that is not part of a budgetary discussion is going to get short shrift for the next several months. So I see that we're going to have some interesting communication issues in terms of, is this important, how is it important, and how can I use it? So those are my remarks.
I think as we did earlier, we'll have all the comments from the commenters and then open it up for discussion. So Dan, you're next in line.
Thank you, Ellen. I'm an epidemiologist, and I wanted to make a couple brief comments. One related to question 1, and mainly an appeal to my own profession that in years past, the great emphasis on epidemiology has been causation and the tendency to report metrics of association, even from population-based studies, as relative risk, and completely ignoring distributions and prevalences and incidences within populations. And as in order to make, if the microbiome concept is able to assert itself into population-based studies, I would encourage members of my profession to think beyond just relative risk and start to think in concepts of prevalence, attributable risk, and those metrics that are of utility to policymakers to understand impact in a population for their purposes. And that's basically all I would say about about that, at least I think. Oh, no, there's one other point about policy. It's a story of analogy, and I don't know whether this analogy will obtain 10 years or 20 years from now, but it's a story back when I was at school at Berkeley. Steve Rappaport was one of my professors, but there was another professor named Bill Reeves who's passed away. And the story is this. Bill Reeves was an expert in arthropod disease, malaria, and the encephalopathies. And after World War II, he developed the sentinel chicken. Now, the sentinel chicken turned out to be pretty important ecological assessment of encephalopathies in the United States, because Bill could go out and harvest from his sentinel chickens not only where they'd been bitten by mosquitoes, but where they'd be infected. Now, years went on, and the destruction of the habitat of the mosquito was so successful that his funding agencies decided that, why are we supporting this program? Because there's not getting any results. Well, fortunately for Bill, and unfortunately for a number of members of the population, St. Louis encephalitis, we had an epidemic of it, and suddenly Bill's work became very relevant, and he was able to sustain his funding. Now, the reason I tell this story is because as a public health person, my goal is prevention. The metrics of success in prevention is nothing happens. When nothing happens, though, it does cost. It does take money to make sure that nothing happens. Bill Reeves' story is an example of assuring that nothing happens and documenting that nothing will continue to happen. Given that that I have a strong sense that the microbiome will depend heavily on principles of ecology and balance, and documenting that nothing is going to happen, an inability to sustain that and to influence our funding agencies to say there is value in documenting that nothing has happened because we are doing the right things to remain in ecological balance will be important. It may not be so important right now, but 10, 20, 30 years from now, it may be very important if this concept takes off and develops as a great integrating issue. I'd also make a comment about is the microbiome simply a potential modulator of risk? It may function in a modulating sense, but I would suggest that in and of its own right, from everything I've heard, perturbations in the microbiome become risk itself, and I think I would leave that to the experts to determine, when is a perturbation of the microbiome going to cause disease in and of itself, and I would suggest that it's like a chain with an equal link in that chain, along with chemical agent exposures and any dynamic that may occur between it. Thank you.
Thank you. I guess continuing around the table, we come to Bill Farland.
Thanks, Ellen. I think this is sort of a seamless transition between our research and policy discussions, and so both because of the earlier discussions and because several of my colleagues have worked with me, or I've worked with them for many years, I'm going to say a few things similar to what has been said already, and perhaps strengthen those themes. The first thing that I wanted to mention was the fact that I think that there's a role for us coming out of this kind of discussion to encourage investment in fundamental knowledge. We did hear that yesterday, and part of this gets at the idea that one of the good things that engineers have done for me is to educate me about a systems approach to dealing with these kinds of issues and the importance of interdisciplinary problem solving. And this is a great example of one where we need to encourage that kind of look at this. This is not something that necessarily is going to be funded by individual institutes or individual agencies, but we've got to find a way to encourage that kind of systems funding. There are a couple ways that we might do that. Certainly federal agencies have a role in encouraging interdisciplinary research through the way that they characterize their requests for proposals, and that's been happening over the last number of years. This is an activity that would benefit by that approach. Currently, we're under discussion about changes to the Toxic Substances Control Act, to the Farm Bill, to healthcare legislation, All of those kinds of things, I think, could benefit by decision makers having an understanding of this concept of the system and some of these issues that could be characterized as public health promotion as well as managing risks. So I think that's something that's worthwhile. Secondly, I want to sort of reiterate the idea that this really helps us in terms of continuing to define what we're calling the exposome. And I think that this is a nice way for us to be able to talk about another form of internal or physiologic type of exposures that we're encountering as we live our lives. I guess I do ask a little bit in that particular context of whether we're talking really about a microbiome or I think I heard somebody use the term functional ome. This question of how we apply the technologies that are emerging to try to get at this issue of are we characterizing populations of microbes or are we characterizing functions of this particular issue, I think is something that is worth thinking about and worth trying to acquaint decision makers who are looking at funding issues and guidance along these sorts of issues. Again, how do we go about doing that? Agencies provide risk assessment guidance in programs and in their research and development activities. This would be a place where perhaps position papers or statements or other sorts of things could move the issue forward. Certainly Journal of Exposure Science and Environmental Epidemiology and Science have taken up some of these issues of the new exposure paradigm and the exposome. Those would be good targets for these kinds of activities to be highlighted for the scientific community. There are a number of other risk assessment issues. I'll go through some of those quickly. I certainly agree that that this is an aspect of pharmacokinetics or toxicokinetics that needs to be taken into account. I would just clarify, I think when we use the term potential modulator of risk, to some degree that's a term of art. I think everybody would agree it's a modulator of risk. In the risk assessment community, we talk about modifiers, and those are sort of addressed after the fact. So you have a discussion of toxicity, and then you say, are there lifestyles or are there other activities that might somehow modify that risk? I think it would be a mistake for us to put the microbiome into that kind of a category. I think it is much more fundamental to the assessment of risk, and so I'll agree with my at least my two colleagues that have specifically said that in this session. Certainly it is a new issue for life stage risk assessments. Again, I think this idea of looking at enterotypes or functional types over the lifespan is something that would help us with some fundamental knowledge. I do think that we could include microbiome data relatively easily in studies like NHANES or the National Children's Study. There are questions that could be added to those kinds of studies that would be relatively simple, in fact, probably quite palatable to people who were responding to those kinds of surveys, so it would be worth focusing on that. I like the discussion that we had about how we define risk and benefit. I think that's particularly important. There are two aspects that I'd like to address. One of them is this idea of the conservation perspective or the ecological value perspective that might be used here. And I want to just reiterate the comment that was made during the discussion that I think if you look at this from the standpoint of ecosystem services, not what an ecosystem is worth, but what it produces in terms of a benefit to society or to individuals or what have you. There's a broad literature out there on that, and there's a real opportunity for us to begin to address some of those kinds of issues. And I thought the discussion about difficulty in getting funding for health goals versus disease was an interesting point that Mildred brought up. And again, I think we can get a lot of traction with this particular issue if we focus on this idea of equal parts health benefit and disease prevention or toxicity management. And then finally, I just put a caution out, and that's that this pendulum swing of thinking of humans as an ecological system and the impact of a microbiome on the way individual humans react to external exposures or disease or other sorts of or pathogens, I think has some benefits and risks associated with it itself. It could have unintended consequences for us in the sense that individuals will look at the uncertainty that this issue brings into our discussion of risk and will basically get into the discussion of the glass half empty as opposed to the glass half full. And I think that's something we have to really avoid. It's important for us to begin to balance this discussion about consistency versus conflicting results and the potential for the microbiome to have an effect, but also that we're confident with much of the data that we've dealt with, and we think we can improve on it by bringing these kinds of things forward. So again, I think that is an important thing for us to deal with, and it's both a scientific perspective and a communication issue that we need to look at. So those are my comments.
Thank you, Bill. Trey.
Well, this has certainly been a very enlightening and just incredible, and I will echo Rita's comments. I think really an eye-opening experience, and I really thank the committee and everyone that has participated for really bringing a lot of great information. I'm at the U.S. Consumer Product Safety Commission. For those that may not be familiar with CPSC, we are a small regulatory agency. We have responsibility over thousands of products that are used in the home. My specific responsibilities, I'm leader for our chemical hazards program, so we're going to look at it from again, from a chemical perspective, as well as the leader for our nanotechnology team, which was mentioned. I think this is a very challenging, exciting time. We have new areas of science, new tools, high throughput screening. We have the microbiome, the proteomics, genomics. So I think it's really a challenge for us to looking broadly how we will incorporate this new information, these new approaches into the way that we address chemical hazards. Typically, we will look at a product and we will try to determine how it will be used. Well, I think one, looking at the product, but also a specific chemical. And I think trying to couch it within some of the questions that are there in terms of the type of research. So with a specific chemical coming from a product, we will look at what is the threshold for uptake into the body, and that's similar to what other agencies do. So one of the questions, and again, I'm kind of answering a question with a question, is should the microbiome, should this be considered? And I've heard that others say that perhaps it should be sort of a target organ, similar to the way we look at the liver or other organs in the body. And if that is the case, then can we, in terms of the research, come up with some type of dose response where we could determine, if this is an endpoint that we should consider, at what level is there harm? And so I think that's where the toxicokinetics come in. So that's really one important point, and I think it goes into two. Is this a target or is some type of modifier? I think the discussion yesterday on arsenic, we conducted a risk assessment on arsenic, and so what happens is how important is the microbiome in terms of activation? Does it increase or decrease the excretion or the absorption? I think these are some very fundamental questions. And again, very fascinating. So I think that also goes into the research. And along with question 3, the ethical, legal, and societal, one of the concerns may be sensitive populations. And I think one of the, some of the information in terms of individual variability, how does our microbiome impact if we look at the modification of the toxicity, what is the variability? among the population? Are there certain sensitive subgroups that we need to pay attention to? So I think that's where 3 kind of is involved in that. So those are the primary issues. Again, I think there are a lot of questions, and I would certainly encourage those that are involved in this level of research to consider these questions as you move forward in your research. That's it.
Thank you. Lita, I think you're on tap too.
I think I'll probably primarily address question 1. I didn't actually take any time yesterday to describe the Human Microbiome Project, but I feel like it's worthwhile to kind of help address question 1. So very briefly, the Human Microbiome Project at NIH is a common fund funded activity for 5 years, started in 2008. Common fund is an internal mechanism for funding programs that are trans-NIH significance. And I wasn't around when that was designed, but very wisely, the folks who designed the Human Microbiome Project decided that the goal would be to produce a community resource. 3 elements to it. 1 is a cohort of 300 adults from 18 to 40 years old with apparent healthy health were sampled across 5 major body sites simultaneously. The skin, the oral, the nares, the gut, using stool as the proxy, and female urogenital tract. Now I say apparently healthy because it turns out that you can be healthy in 2 of 5, 3 of 5, 4 of 5, but not necessarily always 5 of 5 body parts. So they changed the topic to normal cohort rather than healthy cohort. But that's important because the microbiome was sampled and more than once over the course of about 2 years. The data are, I'll explain in a minute why they're not out, but they will be out. The second resource, and again, the resource being to produce data sets that can help catalyze the field. So foundational or reference data sets. The second was Vince Young is part of this. Now we have a suite of about 10 different demonstration projects where the charge is to determine if there's a specific association of a particular microbiome with a particular disease. And in that case, the diseases included, I think, only 3 of the 5 body parts: skin, gut, and oral. I think I've got that right. The third reference, resource was, we were talking about microbes, and so many of the microbes have not been culturable. So we either promoted the development of novel cultivation techniques to get at new microbes, or used, I'd have to call them sort of next generation microbiology techniques, getting at the microbes by actually teasing out a single cell from the community and sequencing the genome of that individual cell. Now the downside is we don't have a culture, but we actually have its genome sequence. We're more than 2 years into the project and we haven't released any data, so that's really bad. But the problem is, or the good side is, that it's a very, very complex data set. And so the group got together to really analyze the data set, determine its sort of quality control, make sure that the data set that gets used is totally understandable about what data set was used for the analysis. And very soon, the Human Microbiome Project will be releasing the 16S data from all those 5 major body sites. This is the normal cohort data. Also what's called whole genome shotgun assembly. So that's the metagenome of those 5 major body sites with lots and lots of information about the QC and other bioinformatic parameters that were applied to a very nice kind of value-added data set. Already many of the microbial genome sequences have been released, and I'm making this point because one really important philosophical goal of the Human Microbiome Project is to have data sets available to the entire community. Now, I want to kind of develop that idea a little bit. I think I tried to give you the example in I think it was one of my last slides where I talked about a scientist at the Argonne National Lab who took data from NIAID, their priority pathogen list, and then blasted it against metagenomes to try to see if there's any signal. I mean, nobody funded him to do that, but he was a very computationally savvy guy who was kind of asking this independent question. And it's been my experience that whether it's the computer generation or whatever it is, there's a whole bunch of scientists out there that have never done wet chemistry or wet biology, but they have tremendous computational expertise and they can get at questions you can't get at necessarily doing wet chemistry or wet biology. And so therefore, to sort of address question one, what I would say, if nothing else, I think a philosophy or a principle that any future study should have is to make the data publicly available. Get it out there, as quickly as possible, because there are entire groups of scientists that can computationally address questions that are going to be very eye-opening to this field. And so that would be what I would want to try to champion here.
Okay, I know we have someone here from the FDA, and I wanted to give you the opportunity to give some perspective from that agency, particularly since I think Rita handed off to you all the hard work related to microbial risk assessment.
Well, thank you. Just as a point of introduction, my name is Carl Cerniglia. I'm Director of Microbiology at the National Center for Toxicological Research. It's in south of Little Rock, Arkansas. I've been involved in this field for a number of years, and I've done a lot of research in what I call system biology approaches, looking at the role of the intestinal microbiota in xenobiotic metabolism. We also have a number of projects getting in line with the NIH microbiome project of actually not only looking at the intestinal microbiome, but we have projects with the Office of Women's Health with skin microbiome, with cosmetic interactions, as well as in vaginal microbiome with interactions with probiotic products. So we have a number of projects that are going on, but more importantly, based on this meeting, is our involvement not only with the FDA, but my own personal involvement at the world level with the Joint Expert Committee on Food Additives, part of the World Health Organization, where we're trying to do risk assessments on a wide variety of food additives, food contaminants, pharmaceuticals, and other products of interest to the world community. So I've been involved in that for a number of years. And some of the points that I'd like to make in relationship to the excellent talks that we heard yesterday and this morning's talk as well is that what I'm very interested in, and my research program is very interested in, and the agency certainly is this whole issue of not only developing methodology to answer these very tough questions that we heard about today, but also it's very important in the validation of these types of things. We haven't really heard too much about that, but it's extremely critical if you're going to use any of this information in risk. And I'll give you some examples where I've been involved in and actually have been doing a lot of research, and papers are either have been published or will be published in this particular arena. First of all, this whole issue of bioavailability, and we heard about it in what Tom talked about yesterday, and Carrie just mentioned it this morning. It's a very critical issue when we talk about food contaminants, food additives, probiotic products, antibiotic residues in foods. As we go down the GI tract, this whole pathway down the tract, the question is do these compounds, what is the true concentration that's bioavailable? How do you determine it? So it's not only it's a big chemistry issue, as well as certainly a microbiological issue, or do these compounds get metabolically active or not? The other issue that I think is very, very important in this, as we go down the GI tract, and sometimes and as we all know, and we heard yesterday, it's a very complex ecosystem, but generally these research approaches that I see are still relatively simplistic in answering these kinds of questions. For example, as you go down the GI tract, you have differences in the concentration of the intestinal contents from, let's say, 5%, 10%, 25% to 50% when you get to a solid fecal mass. And that actually affects the microbial population in what we call phase-associated, particle association, the liquid phase, and there's completely different populations. So if you're doing a risk assessment and you're taking a fecal sample, what is the dose what is the concentration? It's very critical. So we're developing these types of methods to kind of standardize it. Secondly, which I think is extremely important, is actually, and again, I didn't hear this, I'm sure it was probably done, But I think it's very important to do some type of physical chemical characterization of your donor samples as well as a microbiological characterization, because there are, even in a quote-unquote normal sample, there's differences in the water content, there's different in carbohydrate composition, there's differences in pH. pH, if we're talking about drugs, affects the deionization, association constants between drugs, and this affects absorption, desorption off of particles. So this type of information is very, very fundamental in that. Another question that comes up is in getting into this interindividuality or variability issue, which is extremely important, is the number of donors that one needs to sample. Because we heard it yesterday, and these are very expensive technologies, all these omics approaches, and we're using them routinely in our lab from doing our traditional screen. Before, we used to always do the traditional culturing. Now we're kind of getting more into this culture-independent approaches where you do traditional screen, maybe do GGG analysis, and then you get into RT-PCR and pyrosequencing. It's very expensive. So then you have to develop strategies to do this. And then when you get to the transcriptome level, when you get into this very nice functional analysis, it even gets much more expensive. So again, you know, we have to kind of decide how many donors are we really truly going to sample to get a representative data to prove risk. So I think that's another key question another aspect that I think is important, especially with all this bioinformatic information, is what is linking this to public health. And this is something with the WHO we've been talking about for a long time, especially with food additives and antibiotic residues in foods. What is the true magnitude of change to create this? So you see a perturbation, or even you see an uptick in a functional gene or a downregulation of these. What does that really mean to public health? And we talk about that a lot. I mean, antibiotic residues, I've been doing a lot of work in that particular field, and that's a hard thing to really grapple with because, first of all, it depends upon the drug. Every drug has a spectrum of activity, so you can't generalize, you know. I mean, a beta-lactam is going to be different than what we heard about fluoroquinolones yesterday, et cetera. But also, when we talk about resistance, and this gets into screening your donor samples, we all have background levels of resistance. The genie is really out of the bottle in terms of that. For fact, tetracycline is an example where you may have 80% of our samples are highly resistant. So if you're using tetracycline class compounds of drugs, what is that magnitude of change in resistance to show that it impacts health? You know, so it's important to really screen these samples quite well, especially when you're talking about antibiotics, to know, because we all have background levels. So we've been doing, you know, a lot of work, and I can keep going on and on, even in standardization of media. You know, it's very interesting, and getting back to the example that was used this morning where the literature was surveyed from a public risk perspective. Well, I've been surveying the literature quite a bit to see how samples are stored, which affects all that is can affect diversity. How samples are, the media that's used, you know, in growing these, which also affects these things. So we're kind of looking at a lot of these parameters to look at, to really get some more validation so we can be a little bit more comfortable with the data we're doing. So that's kind of a short summary of what we're doing.
Great, I'm glad I asked you. Overall moderator, what is our plan now in terms of time? Obviously, we'd like to have some time for, I don't know if that's Helmut or who down at that end of the table. In terms of time management at this point, we're supposed to close at 11:55. Obviously, it'd be useful to have a discussion of the comments made by this panel.
I have a semi-burning comment, and it's, you know, Lita, you talked about sharing of data, and I just wanted to strongly endorse that. Now, from an omics standpoint, you know, we've all been doing that ever since we started publishing, and we journal editors really enforce that people put their raw data into some sort of publicly accessible database as a precondition of publication. But a lot of what we're talking about goes beyond the microarray kinds of experiments, and I think that that's worth just putting on the table as something that's going to be important as we go forward with computational approaches. A lot of the sort of the epidemiology kind of data, maybe even the animal study raw data that is a lot less, sort of resource intense in terms of storing, but has been held a lot more close to the chest by people, I think needs to also become a public resource in order for us to really make progress in these kind of genomic, meta-epidemiology kinds of questions that we're talking about addressing.
I would just like to add, based on the last comments from Carl, that we need to explore what kind of annotation is needed. I'm not sure if that has been settled. And as you know, that was a topic of intense discussion, which the NAS helped facilitate on toxicogenomics. But just to put on the table is something for the HMG to think about, coming to some consensus view of the annotation of these studies so that we're all certain the variability isn't being introduced by differences that we have perhaps not laid out on the table.
May I comment? So actually, this has happened. This grassroots activity has happened without our directing it, because this computational community really wants to be able to compare apples to apples. There is a group that self-formed called the Genome Standards Consortium that has tried to really champion. I mean, it started out obviously with having common data terms and standards around metadata for genomic data, but it's grown beyond that. It's gone into market data and metagenomic data and so on. So because there's so much computational expertise growing out in the biological sciences, there's been this very strong community level interest in developing community embrace standards for how data are reported so that
I just want to be clear. My concerns go beyond that. It's really annotation of how the studies were done.
Absolutely.
In terms of being able to ensure that, say, if I repeated the studies that Les did, I get some different results. It's not because I did something different in my methodology. So not simply reporting, clarity and consensus-based reporting of data, absolutely, but also I'm really reflecting your comments. And I'm not saying we need to settle that here, but just to note it as something the field needs to come to agreement on.
Of course.
Brief observation, as we're talking about policy, and this is spurred by Dan's comment in the light of Catherine's enlightening discussion yesterday, which is that we should be, as we think about sustainability and what does sustainability mean, we have not been thinking about microbial sustainability. And when we talk about the sentinel chickens and kind of looking at the ecosystem indicators, just that whole concept of the multi-generational nature of the human microbiome and the way we have to keep incorporating it from our environments, we should be incorporating sustainability of that of a healthy microbiome in our concept of sustainability.